As we turn our attention from replication to transcription, I've decided to stay with our old school technique. I don't have my sister's awesome drafting paper, but I think we can make it work. So recall that transcription is the process of converting the DNA code into RNA. Let's begin by looking at the two strands of DNA. There's a lot of terminology that surrounds transcription. So if we look at one strand of DNA, by convention, we typically write the 5' to 3' strand on the top, and then we write the 3' to 5' strand on the bottom. Now, really, we understand that this is just all a matter of perspective. We could stand on our heads, do a yoga pose, and see that the red strand was the top strand and the blue strand the bottom strand. So we just have a lot of conventional terminologies that have come up around this. And we'll see in a moment that a lot of how we draw the orientation of the top and the bottom strand is dictated by the location of the start site of a gene. And so on this particular construct, let's draw a region here that we know in actuality would be a coding region, a particular nucleotide sequence, but we're just simply going to call it gene 1 here. And we're going to recognize that there is some unique sequence that occurs next to this gene that dictates the start of that gene. So I'm just going to show this as an orange box for right now. So a lot of terminology has come up around this. By convention, we call this top strand the coding strand or the plus strand. So this strand here is termed the plus strand or the coding strand. And then likewise, the bottom strand, as you might guess, is called the minus strand. Just going to write that out, minus strand. Or it's also called the template strand. Now, that's going to make a lot of sense when we realize that the bottom strand is the strand that gets read by RNA polymerase when RNA polymerase converts this code into RNA. But for now, let's make note of something else. The gene is said to begin at what we call the +1 transcription start site. Now, that is just conventional once again, because that is the way in which we have numbered the nucleotides. So the nucleotide that is the first nucleotide in the gene is called +1. So the nucleotide to the left of that is going to be called -1. The nucleotide to the right of that is going to be called +2. Now, this region that I have shown in an orange box here, this region is said to be upstream of the gene. So if we would just say from everything to the minus side of the gene is termed upstream. And everything to the right of the +1 transcription start site is downstream of that site. Or we might just simply say that this whole region beyond the gene, to the right of the gene there, is downstream of the gene. So we could call this region the downstream region. And we use these terms a lot. It's just like a map again, right? It's like the map of the gene construct world. And we want to be able to refer to those areas that are both upstream and downstream of a gene. Or sometimes we just want to refer to those nucleotides that are either upstream or downstream. So we could get really discerning here and say that the -1 nucleotide is upstream of the +1 nucleotide. There is no zero. Or we could say that the +2 nucleotide is downstream of the +1 nucleotide. So we could get very, very discerning. And this just allows us to orient ourselves, to think about geography of a gene. Oh, that's cool. Geography of a gene. Right? So now let's think about what happens when this gene the code is converted from DNA to RNA. So what will happen then is that this epic enzyme called RNA polymerase will bind right here at this region just upstream of the +1 transcription site. Now, in order to fully relate geography, we really ought to name that. So this region here that I've been showing with the orange box, why this region is termed the promoter. And the promoter is the nucleotide sequence to which that RNA polymerase, the purple blob that I've drawn there, recognizes. So it binds to and recognizes that sequence there. Now, when RNA polymerase recognizes that sequence, RNA polymerase, unlike DNA polymerase, does its own unzipping of genes. So we say RNA polymerase unzips its own genes. And so it actually opens up a region right there at the promoter. So let's try to give that a bit of drawing. So if we were to show a little bit of time lapse, and I think my kitten's going to help me. You've all met Ripple before, right? RNA polymerase will actually, when it binds, will actually unzip a region somewhere around 16, 18 nucleotides worth of the DNA and leave it denatured. That is, the hydrogen bonds between the A's and the T's and the G's and the C's are broken. So now our RNA polymerase is bound here. Now, in fact, this is actually called initiation. That makes sense, right? Because it's the start of everything, as the promoter region is recognized by that RNA polymerase enzyme. And as I said, it denatures about 16 or 18 nucleotides worth of the DNA, unzipping its own genes. And RNA polymerase it's a very, very complex quaternary structure. In fact, it has, remember that means it has multiple subunits in it. Alpha, beta, beta prime, sigma, and omega. We're not real clear on what omega does, but we do recognize a very important polymerization role of the other subunits, and sigma plays a very, very unique role. In fact, sigma factor, and I'm going to give it a cartoon depiction, maybe just showing it here in black. And let's just say that sigma is sort of the eye. Sigma factor really recognizes promoter sequences. Sigma factor gets to a promoter sequence and says, oh, hello, good looking. And it slows everything back, and it binds really specifically to promoter sequences. So we can just say that sigma factor recognizes promoter sequences, and it has a much higher affinity for promoter sequences than it does for any other DNA. And so it really slows RNA polymerase down when it hits a promoter. Now, once this occurs, this opens up this short stretch here of DNA for RNA synthesis. What we can recognize now is that RNA can get started being built. Now, this is where this term template comes in, because this bottom red strand is going to serve as the template for building the RNA. So that means that the RNA is going to pair with the red strand here. So at this point, once the RNA polymerase has opened up this region, there's going to be the possibility for synthesizing a very short stretch of RNA that is going to pair with the template strand. Now, this is happening inside of the RNA polymerase enzyme, so it's really quite a very short stretch. And I could probably show better that that's inside of the RNA polymerase enzyme. Make our RNA polymerase enzyme a little bit bulkier there. It's actually occurring within the RNA polymerase enzyme. Now, remember that RNA polymerases, unlike DNA polymerases, are capable of starting cold. So they just kick off the synthesis of that small region of base pairs, 9 or 10 nucleotides inside of the RNA polymerase enzyme. Now, as you might guess, just as DNA polymerases, they synthesize only in the 5' to 3' direction. So inside here, where we see the 5' end being the first little end here, that after it gets synthesized is going to escape as this RNA polymerase heads down the construct. And inside here, we'll say this right here is the 3' end. So as RNA polymerase builds, it's building onto that 3' end. So 5' to 3' synthesis. Now, we are ready to see this move into the elongation phase from the initiation phase, and to build that RNA even more into a longer molecule. So if this is initiation, then we got elongation next. And this is somewhere mid gene 1 now. So it's in the gene. And the RNA polymerase has now cleared the promoter. Now, let me tell you, sigma factor doesn't want anything to do with an RNA polymerase that has cleared the promoter. So sigma factor gets the heck out of Dodge because it only has that strong affinity for promoter sequences. We actually call it a promiscuous subunit. It's going to go off now and associate with another RNA polymerase enzyme. But now the RNA core polymerase enzyme is in the elongation phase, where now it's reading the coding region of gene 1. And of course, at this point now, the RNA is beginning to elongate. So, recognize that the first 9 nucleotides that occurred here were those that were just in the beginning of gene 1. So they were just the first 9 nucleotides in the beginning, because of course +1 is where the transcription start site happens. So this is just the very beginning of gene 1. So now we see that this RNA is now, as the DNA comes back together after RNA has melted it, denatured it, and moved along, the RNA gets pushed out. So the 5' end now is just peeking out, because now the RNA polymerase is moving down gene 1, and the RNA is getting longer, pairing with the template strand of the DNA. So we need to do more time lapse here. So now the RNA is getting quite long, and the 5' end is fully sticking out. We know in here is that 3' end from which the onto which the new nucleotides are getting added as that RNA polymerase heads towards the end of the gene. And of course, eventually, Ripple's showing you there'll be a transcription termination site down here at the very end of the gene. So then we'll see down here at the end of the gene a transcription termination site. And so the last stage, we go initiation, elongation, and then finally now we're about to head into termination. And let's think about what can lead to termination. And there's a couple ways in which termination can take place. So here's our RNA. It's still got that little transient pairing inside of the RNA polymerase, but mostly it's completely made, almost entirely done. But now what's happening is we've hit a site within the sequence of the template strand that has in some way encoded for the end of all of this. One way that can occur is actually something called the hairpin loop formation. So let's draw another image showing this hairpin loop. And what causes this is that in the RNA, there is a region in the sequence of the RNA that is complementary to itself. Like, say, for example, there's a whole bunch of A's followed by a whole bunch of U's, and this creates in the mRNA this hairpin loop region where it actually binds back to itself. So there's hydrogen bonding occurring here in this loop, and that destabilizes the RNA DNA heteroduplex. Okay, so that's the fancy name that we call what's going on here in the RNA polymerase enzyme is an RNA DNA heteroduplex, meaning that it's one strand RNA, one strand DNA forming that region there. So this hairpin loop destabilizes that and leads to termination. Another thing that can happen is instead that an enzyme might bind called Rho, the Rho enzyme, it's an ATPase, can get itself all wrapped up in this RNA and actually destabilize the heteroduplex. So one thing that can happen is the hairpin loop formation. Another thing that can happen is termination via the Rho enzyme. So I'll draw that. So this is the Rho enzyme here. Really what happens is that a region of the RNA kind of ends up wrapping around this Rho enzyme. And really Rho enzyme just kind of wraps itself up and pulls and then sort of destabilizes the heteroduplex, obviously. So Rho dependent termination is this form of termination. So this enzyme is Rho. I'll write it in orange. Okay, so those are the two ways that termination can take place. Now, as we've finished this off, we're starting to see something that is epically important. In fact, I will probably re-emphasize this point utterly dozens of times, and that is that as we've seen the formation of this mRNA, it has built based upon the code of the template strand, the bottom or minus strand. So when it's paired with that template strand in order to form, it means that this mRNA or RNA of whatever type now has the same exact sequence as the coding strand, right? Because it too is complementary to the minus strand. So the mRNA looks like the coding strand of DNA, but it's RNA. So we see that NTPs were used to build it rather than DNTPs. So we know that that 2' position has a hydroxyl instead of it being deoxy. But then additionally, we know that anywhere that there was an A in the template strand, that paired with a U rather than a T in the mRNA. So it looks like the coding strand, but U's replace T's, okay? So if we wanted to do a zoom in, which we probably should do, on what's going on within the heteroduplex, so let's see if we can make a zoom in here. Let's give ourselves a sequence here. So if on the template strand of DNA, we see T, G, C, A, okay, if this right there is what is the sequence that we see, we're seeing this sequence here, if that's that sequence, then the RNA that's temporarily paired inside of there, it's building onto the 3' end, so it has paired a G with the C, right? It has paired a C with the G and an A with the T. But here, as it adds now, right, here's the 3' hydroxyl right on that G, and it's going to add a U right there. So this is the most 3' end that it's building on, and that's what's going on in that temporary pairing right there. So that's that zoom in, which I probably should have moved up a little bit, but that's what's going on in that zoom in. So we're recognizing that this mRNA looks very much like the coding strand. So when we're reading in literature, we're often going to see that we're just going to see a long sequence of DNA that is just the coding strand. And from that, just knowing the sequence of this coding strand, we need to be able to both determine what the template strand would look like, and we need to be able to determine what the mRNA would look like as well. So we can easily make that leap now that we better understand what's happening in transcription itself. Now, there's a couple more things that we need to think about sequence-wise. So if we return back to our earlier drawing, where we met the promoter for the first time. So recall that we realized that the promoter is actually a sequence of nucleotides. This nucleotide sequence is what sigma factor recognizes. Now, in prokaryotic systems, that ideal consensus sequence is known. By comparing promoters across organisms, they have found that there is a sequence that occurs the most frequently in 2 areas. Now, these 2 promoter sequences are found at minus 35, often called the minus 35 sequence, meaning that it's 35 nucleotides upstream of the +1. And the second one is the minus 10 sequence, which is known as the TATA box. So these 2 sequences, when they are the most commonly occurring nucleotides in those positions at those sequences, at the minus 35 sequence, it is T, T, GACA. At the minus 10 sequence, it is TATA at. Now, if a promoter has the exact sequence TT-GACA and TATA-AD at its -35 and its -10, then we say it is an extremely strong promoter. Now, if the promoter differs from that, then we would recognize it as becoming weaker and weaker as a promoter. Now, we're going to practice that in class, so recognizing the closer to that ideal sequence, the stronger the promoter, the farther away from that sequence, the weaker the promoter. Now, what does that mean? Well, that just means how frequently does RNA polymerase bind to and recognize, or specifically the sigma factor, bind to and recognize the promoter sequence. The stronger it is, the closer to ideal, the more like this sequence it is, the more frequently the RNA polymerase enzyme binds and reads the downstream gene. Now, in this case, we've shown only one gene that is under control of this promoter. So when the RNA was made and we got this final RNA, and in fact, let's go to our final product, our final RNA. This mRNA would carry the information for just one single gene. When everything is said and done and that final transcript is released after the termination, we call this a monocistronic mRNA, meaning that it's carrying the information for a single gene. But in many cases, in bacterial systems, we see a situation where we have a DNA construct that carries the information for multiple genes, or said another way, where there is a single promoter that controls the expression of multiple genes. So here's our single promoter. And then downstream of that promoter, we see gene 1, gene 2, gene 3, maybe gene 4 and 5. For example, in the construct that encodes for the biosynthesis of tryptophan actually encodes for 5 different genes that, of course, unsurprisingly, encode for the production of enzymes involved in the anabolic pathway of tryptophan biosynthesis. So we can see multiple genes all under control of a single promoter. This has a special name. It is called an operon. So an operon, when it is expressed, that's a fancy term for when it is converted from DNA to RNA into protein. So what will happen when this is turned into RNA? We know the RNA will look like, it'll begin right here, right at the +1, and it will look like the coding strand, and it will carry the information for gene 1, gene 2, and gene 3, and we will call this a polycistronic mRNA, carrying the information for more than one gene. Let's end today by adding a little bit of complexity to all of this. I've drawn a plasmid. We're going to call it P-Fun, just for fun. The P-Fun plasmid. We know that this plasmid is going to have its origin of replication. And on this plasmid, we're going to say that there are 2 genes. We'll begin with gene 1. And here is the promoter for gene 1. And we're going to have gene 2 as well. Here's the promoter for gene 2. And so here's gene 1. And here's gene 2. Okay. Now, hey, notice that these are not operons. They have a single gene controlled by its own promoter. So we know the RNA, the resulting RNA, will be monocistronic. But let's think for a moment about how it is that everything is relative on a gene construct. So when RNA polymerase binds and transcribes gene 2, it's going to head on down, going from the promoter down the gene and reading the template strand. Okay, clearly that template strand is this red strand right there. That is going to serve as template. And as the RNA polymerase reads, it will make an mRNA that looks like the blue strand and carries the information for gene 2. Okay, a monocistronic mRNA looking like that blue strand, only use replace T. So here's our monocistronic mRNA. Now, let's think about gene 1. RNA polymerase binds, unwinds, and reads. Whoa, check it. As RNA polymerase reads, it reads the template strand, which in this case is the blue strand, and it synthesizes the monocistronic mRNA that ends up looking like the red strand, only use replace T. So this RNA looks like the red strand where the blue strand was template. So as we situate ourselves on our PFUN plasmid, we recognize that everything has to do with perspective as to which is template and which is coding.